So many of you who attended this conference in the past years have seen many of these slides, but then I still hope you will find some new information as well. First, my disclaimer: I'm employee of GSK PLC, and in this presentation, the opinions are my own, not that of the company. What I'm going to talk about is, as usual, I give an introduction of how genetics and genomics can help study, well, glycobiology in particular. In fact, any biology, it's kind of this wide, in a way, holistic method which is applicable to a very wide range of problems, glycomics in particular, and then talk about what we have learned about genetic control of protein N-glycosylation in human populations, combined methods of human quantitative genetics, and finally I have some contemplation, conclusions, discussions, summarizations. That's probably the interesting part, right? Okay, so the problem is that if you want to study human biology, it's a terribly difficult thing to study, right? So you cannot really do, like when you have model objects, right? You can do really good experiments. With humans, that's not the case. The only truly good experiments you can do would be clinical trials, double-blind, and this is super expensive and super slow. All other things are not living up to true experimental standard of true experimental science, right? So humans are a terrible object to study. If you're a biologist, probably you really want to avoid studying humans altogether, right? You study mouse or a worm or something with very short life cycle, right? So what geneticists said, for hundreds of years, you better study a fly than an elephant, right, or a human. Now the thing is that, of course, eventually we are all very interested in human biology, right? Because we are humans, right? And we have diseases and we die, right? And we want to learn how to cure these diseases and how to extend our health span and lifespan, right? But then we are back to square one. Humans are a terrible experimental model, right? But biology of human is not exactly the same, right, as biology of a worm or a mouse, right? Now, what's good about human genetics and genomics is that that's about one of the, and the only tool, right, if you forget about clinical trials, that really allows you to study in vivo human biology, right, by piggybacking on experiments of nature. Right? So, what are classical genetics approach to study model organisms? You knock out a gene or you overexpress the gene and you see what happens, right? Okay, you cannot exactly do this in humans, but you can look into what nature have done for us. You can look into what loss of function variation, which you see in human populations, does to the phenotype, right? What a variation in some enhancer element, which is tissue-specific change, this tissue-specific change in expression does to human phenotypes, right? So in that, you are not able to do experiments yourself, right? But you fall back onto what you can see in nature, kind of experiments of mutation process extended by the drift, etc., 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 right? So this is, if you want to study in vivo human biology, genetics is one of the options. Okay? Now, a good thing about a genetic approach to study biology is that essentially you need to have only two things. You should be able to measure genomes as to see what are the genomic variations between different people, right? And the other thing, you should be able to measure the thing you want to study. You should be able to measure the phenotype, which is in fact not so obvious. We are very good in measuring genomes, right? We now can sequence hundreds of thousands of genomes in a matter of months. We do it with very good precision, right? It's totally reproducible, right? Phenome, and when you measure the genome, you know what you measured and how much is missing, right? Talking about phenome, what is phenome? When do you tell it's measured? It got the space dimension, it got also temporal dimension, right? So in that, phenome is way more difficult thing to deal with. But anyways, as soon as you can measure these two things, you can apply genetic tools, right? You start looking into how variation in your genome translate into variation in the phenotype. And there are, many complicating factors, but the basic idea is, very old and works pretty well. Okay, now of course, the thing is that if you are doing experiments, right, you can knock down, knock up every gene, right? When you observe what nature kind of have done, you are restricted, right? Because you are, if you're interested in the area of core biology, some processes which are essential for life, right? You don't really expect to see mutations in some genes, right? If mutation in some gene is unconditional, dominant, lethal, you will not see it in an adult human, right? So your visibility when you are doing in vivo human biology is somewhat restricted to this more peripheral biology where the genes, when mutated or their expression is changed in tissue-specific or non-tissue specific manner, that organism can tolerate it, right? So you can see these effects in a human. Okay, so the interesting thing is that when you think of like this classification of like biological process, and this is a bit vague term, but maybe it will become a little bit more clear when I talk it specifically in application to glycosylation, is that this core biology and like more peripheral biology, right? And what you expect is that your core biology is not really tolerant to mutations, right? You would expect it to often be evolutionarily conserved, right? Because it kind of evolves very slowly, right? And if you observe mutations there, they are probably quite rare. They are probably leading to quite severe phenotypes, right? Now, this peripheral biology that's where species evolve, right? There you expect to see, a large population of variations, and sometimes even, knocking down this gene doesn't lead to any, visible effect on the phenotype, right? FAT6, famous example, one of the famous examples, actually. Many of the terminal glycosylations are quite tolerant to loss of function mutations, at least in heterozygous form. And yeah, so this is kind of global picture. Okay. Yeah, right, sorry. I'm doing so much hate invading. So, right. So, what tools do we use to study human biology with quantitative genetics? Right. So, one of the main tools in genome-wide association study, and in this study, you are trying to, as I mentioned, correlate variation in the genomes of the people with the variation of the phenotypes. And what you figure out are statistically significant the regions of the genome which statistically significantly associate with a phenotype of interest or a set of phenotypes of interest. And I will skip this detailed explanation how you get to genome-wide association study, just showing the final result, which is often presented in the form of Manhattan plot, where on x-axis I have my genome, different chromosomes colored in different colors, and these dots passing the lines basically indicating the regions of statistical significant associations, right? Because the problem is that in the genome, okay, it's linear structure, it got dependencies, linkage disequilibrium, right? So when you get a signal in the genome, like this here, pretty strong, right? When you look down, there is a whole bunch of genes. So there is this problem of like telling, so function of which exactly gene is affected by the genetic variation in this region. And if you are kind of lucky, in this gene you will see a coding mutation, something like that, which makes you pretty confident this is the gene. But most of the time what you see is regulatory variation. So it's probably variation in some enhancer, right, or outside of core promoter region. So you cannot really unequivocally relate your variation to which exactly gene's function is affected, okay? So it's like a big deal in my field to try to solve this, right? Okay, Now, in my group, we've been for a long time interested in these two questions, like which often tissue-specific network of genes determine inter-individual difference in human protein and glycosylation. And I think this is the second question, but many, if not all of us, are interested in what is the nature of association between protein glycosylation and complex disease, right? Okay. Now, how we studied, I mentioned that we are very good in measuring genomes. Phenome, in this case, thanks to the work done, Pauline, Gordon, Manfred, right? We can measure pretty well and at large scale. Scale is very important if you talk about quantitative genetic applications. You need thousands, tens of thousands of people with genomes and the phenotype to have robust links and to replicate them, right? Again, like Gordon said, everything needs to be replicated. Everything I mentioned here was replicated, right? We don't want to spend our life working on noise, right, and making fantasies about it. Okay, so this is our phenotype, right? And we basically reduce this complex chromatogram for a single individual in a number of quantitative traits. So that's basically the height of the peak, which quantifies the relative abundance of well, essentially often not single glycoform, but often it would be like the leading glycoform in that peak. Okay, well, good enough. Right, and this is a slide I demonstrated last year, and at that time we've been to, in total, 55 replicated loci for association with, well, different singular proteins like IgG and transferrin and glycosylation, but also blood plasma, which is like the totality of glycosylation of blood plasma protein, right? Okay, now what changed? We are, well, to +10 here and +8 here. Why the difference? Because things overlap between, you know, different domains here, right? And this is the work published in bioRxiv, and well, that's, I think, something we did with Manfred, who's sitting on the first row. We did something, I think, very interesting, in that we were able to combine the data from mass spectrometry and UPLC, right? And they are not measuring exactly the same things, right? And the last time we were trying to combine this, it's a paper we published with Gordon a couple of years ago, where we basically selected, I think, 4 or 5, well, a little bit more structures, where we can kind of very confidently map the mass spectrometry peak into the UPLC peak and other way around. But it's very, very restricted, right? Now, what is interesting about this work, well, one of the aspects, right, there are a few more, is that we applied a new statistical method which would allow us to meta-analyze all this data together, right? And again, like, interesting thing here is that I think most of us think about statistics as something, you know, 500 years old, well, if not 500, maybe 100 years old, right? People like Fisher introducing things like t-test. It's something is immutable and not changing, right? So what we've been using here is a new test, Cauchy aggregation test, which was described in statistical literature only 5 years ago. So this is like brand new math. You don't expect that often, right? You expect a lot of like brand new, you know, biotech, but brand new math. That's pretty cool. Okay, and it's also like touch upon the problem that we have different platforms, but even within single platform, your resolution evolves. You get into better resolution over time. And Gordon mentioned, like, okay, we are now to 200 by 250,000 of glycomes, right? But what are the meaningful ways to put all this information together, not by, you know, slicing this or that, you know, peak, right? Or, you know, because she is good, it's still a little bit rough method because it doesn't really tell you, like, what exact structure is associated, right? It's just, you know, mounts the statistical evidence saying, like, some of these glycans is associated here, right? And then, okay, of course, you can try to dissect it. But still, interpretability is hard work. I'm not saying it's not interpretable, but it's hard work. Okay, Now, if we try to look into, so this is basically the material. As geneticists, right, this is what you say, so this 63 loci. What I can learn about biology of the trait I'm studying from this, by looking into this. Can I do some predictions? So nice thing about genome is that so many things are linked to the genome, the proteins, the pathways, other disease, right? So once you map your things on the genome, you can go into so many directions trying to understand what's going on, right? So this is our starting point, your 63 genes, okay? Now, what we know from biology, not from genetics, right, is that if you look into glycosylation of total plasma, right, you would expect this to be a superposition of the glycomes of proteomes produced by mainly two tissues. That would be your liver-secreted proteins, and this will be your immunoglobulins. Okay? Now, this predicts, and it's kind of what I'm showing next is kind of trivial, but it's trivial to some point, right? Because I'm saying, okay, this is what I expect. If I look into the data, do I see it or not, right? And when you see it's trivial, right? But the thing is that when you start checking trivial things, often they are not exactly matching, and that often gives you a clue that, okay, maybe it's your data is not perfect, but maybe it's your understanding of the problem is not perfect, and that's an interesting piece. Okay, so from this picture, you would expect, if I'm looking into these 63 genes, right, I expect them to be expressed in liver or in antigen-producing cells, right, well, bone marrow, most possibly plasma cells, okay? If I'm doing this, I indeed see this picture, and I also nicely see this gradient, where on the top I have genes which are mostly expressed in plasma cells and have low expression in liver, right? So I would expect that, well, I will tell what you expect later on. And then on the bottom I have these genes, and you can see things like CFH, complement factor H, and antitrypsin down there, which are expressed mostly in liver, right? And there is a bunch of genes which are expressed well in both tissues. Okay? Now, what I can also do, I can look whether these genetic regions are associated to certain spectrum of glycans. So don't try to read this too much, but basically these are genes linked to the glycan structures, which they are statistically significantly associated, and then these glycan structures are painted based on, like, okay, this is what we expect to see in liver-produced proteins, right? And this is what we expect to see on IgG. So again, like, this is phenotypic, glycan phenotypic spectrum to put these genes into two buckets, right? So here we look at the expression, 2 buckets, plasma cells, liver, right? Here we look into their phenotype, right? 2 buckets again. Okay. Now, if I put this together, does it make sense, right? And like 80%, 90% maybe it does, right? So I see that the genes which are expressed highly in liver, but kind of have very low expression in plasma cells, right? Their phenotypic spectrum is mostly about, well, it's 3-ternary, tetra-ternary, things you only see on liver-produced proteins, right? So, well, kind of good. This area also is good, right? So these are genes which are expressed highly in plasma cells, almost not expressed in liver. So blue is the phenotypic spectrum of glycans, right? So prime and ternary, these kind of things. So like, okay, it's so far so good. This is probably also not a big problem to see things here. But again, like, Manfred, we talked about that yesterday, like consistency, right? What you strive for is like consistency, and not 80%, preferably like 99%, right? So if I start looking in details, I start asking questions. For example, you have ABO here, right? And ABO has nothing to do with the spectrum of glycans we are looking at. Moreover, it's not really expressed in these two tissues. So the mechanism of action is some very different from, plain idea that, this is a glycosyl, like this one, right? This is a glycosyl transferase. It's expressed in liver, right? When proteins are produced, it puts sialic acid on them. It works better, more sialic acid, right? Very simple. Right? ADO definitely works in some different way, right? And there is this B3 gut story, right? And I think we did figure out that you do see these structures indeed in human plasma. But again, where does it work? And it may be a problem with the data, right? So maybe our expression data is not good, right? We need to dig more into this. But maybe it's like tantalizing biology. It may be experimental artifact, right? You don't know. It makes these things very difficult, right? I'm not talking about B3 gut and your work, sorry. I'm talking in general, of course, right? So, if you look in the global picture, that's kind of, so far so good, right? You start looking in details, you start finding the spots where you start asking questions. Every spot which you start asking questions may be worth, years of work and full-size PhD. Right? Maybe not, because it could be artifact. Okay, and then if we look, I mentioned nice thing, you map things to the genome, you can look outside, right? Because so many things are mapped to the genome, and we looked at the genes which we see involved in the protein N-glycosylation changes, right? They are also involved in a very big spectrum of other traits, right? And this is the GCKR is very pleiotropic, right? It's it's associated with so many traits, including N-glycosylation, right? And then there are many others with tens of associations. And if you start thinking of this, you really don't know how this works, right? And then you can hypothesize about a couple of genes here, what may be the mechanism of action, and you make a step or two, and then you're often stuck because things stop being consistent again, right? There is a little consistency, and then again, you cannot, it's hard to achieve full consistency. Okay. Now I'm going back to bigger picture, even bigger picture now, and saying, so this is our expectation. If we have core biology and we have kind of periphery, right, we should expect this gradient of tolerance to mutations, potential evolutionary conservation, and to what kind of phenotype the changes in these genes induce, right? Okay, so what we did, we took this, this is N-glycosylation pathway. These are early stages, right? And these are the stages where we get to this structure, which is common for all complex glycans for mammals. And we split basically these genes in three buckets, saying this is very early stage, this is medium stage, this is late stage. I think when you think what is core biology here, so probably these two stages are core biology, right? But you can argue also things here could separate, okay? So, but basically we put these genes in three buckets and we start looking into different characteristics which occur in these three buckets. So first thing, if I'm looking into congenital disease of glycosylation, which is very severe syndromic phenotype involving developmental problems, this and that, really classical syndromic stuff, right? I can see that, and note here are 13 genes, here 13, this is twice as much, 28, right? So I do see that the congenital disorders of glycosylation, the genes which when mutated lead to them are concentrating on the early stages, right? So that's kind of makes sense. That's my expectation. Core biology, more severe phenotypes, less tolerance. Okay. Now the same is with when I'm not looking specifically at things classified officially as congenital disease of glycosylation, but I look into like multi-system syndromic things and things involved in developmental problems, right? I still see the same gradient. Okay, so again, like this is like the part which you don't want to mess up with, which is like trivial and obvious in a way, right? Still, if I look into where glycosylation Q-tails map, right? So the work we did on populational variation, right? The things we see like variations in, you know, each of, many of us are heterozygous for the variation in these genes, right? So this is like very, very common, right? And you see the phenotypic variation all over the place. They are exclusively concentrated in these stages, right? This may be a bit of artifact, though, because there is this thing about a complex phenotype, but specifically also about the biochemical pathways. Things get buffered. So if you mess up something in early stages, you often don't see the effect much further down the line, because biochemical pathways, unless you really like kill the reaction, if you just reduce the rate a bit or by 50%, it doesn't change. It doesn't change the end product of it. Right? So there is this whole, you know, capacity of buffering and compensation, right? Okay, and finally, if I look into the tissue differentiation, right, and I use for that this tau index, which is kind of entropy, right, I see that there is way more tissue specificity in expression of the genes at the later stages. So I can almost speculate, like I'm seeing here, I'm seeing kind of evolution in action, and that's probably the way the tissue specificity evolves from, you know, first populational variation, and then it may get fixed, right? And then another mutation come, and you can like suppress expression in this tissue to zero, right? Or you can pump it up, right? So it's you know, it's very, it would be interesting to speculate on this, but I'm not sure it's exactly what we see, because again, like buffering, it may be few things we see are artifactual in a way, or like interpretation should be different. What is interesting, and I told you, like, well, I'm going to check trivial things, right? So when you see these things, it's like people tell you, but it's obvious, right? Now, the interesting thing, if you look into loft tolerance or loft intolerance, we didn't see anything, right? And then we look into like evolutionary conservation of these genes. We don't see a big trend, right? And with this, I may think that what we did was really rough, but also you do rough things. If the effect is very strong, you still expect to see it. With loft intolerance, it probably tells about metric we are using, because again, like if you look into these diseases, they're mostly recessive, right? And then this metric is characterizing way more dominant or semi-dominant things, right? So this is telling me about my tools potentially, right? That I don't have a proper tool to assess it, in fact, right? Maybe. Again, this is not obvious. When you don't see an obvious thing, it's totally not obvious why you don't see it, right? Okay, I'm going to summary, and so, well, that's all news. Plasma N-glycosylation is driven by lymphoid and liver glycosylation, right? We see a lot of links between, you know, genes involved in glycosylation and all kind of disease, right? It's very not clear yet how this works, right? And we think often the mechanisms are not direct, right? Again, like you see this ABO, and ABO is associated with pretty much everything, right? But how ABO works on glycans, it's which we measure, right? It's, you know, it's not direct. Syndromic things are concentrated at the early stages for glycosylation, well, trivial. Genes encoding terminal glycosylation show high tissue differentiation of their expression. We don't observe, with what we did now, relation between stage of N-glycosylation and evolutionary conservativity or loss of function intolerance. Probably telling about our tools and how we use them. What is interesting, and I didn't talk much about that, is genetic control of population variability, right? So, and it's about this, it's almost tantalizing to think that you see evolution in action, right? So, as you go to terminal glycosylation, you see increasing tissue specificity, right? Some tissues get off, you know, some on, and you can understand like why you can allow it, right? But if you look into variation, it's tissue exclusive as far as we can see, right? So genetic control of variation of terminal glycosylation is even more tissue specific than tissue specificity of the expression at the same stage, right? So again, this kind of tantalizingly suggests that, well, are we seeing evolution in action, in fact? Okay, but that's clearly a speculation. Okay, I mean, the big question is how protein glycosylation relates to pathogenesis of human disease, right? And me being geneticist and genetics and genomics being these tools which kind of one size fits all, right? And that's their strength and power. You can apply it so many things, right? But it's also, of course, the weakness because, again, like this big picture, oh, it's 90% consistent. Cool. What about this 10%? Right? So the way of doing this kind of science is to dig wide, right? You dig wide, well, a little bit shallow, right? And MOA is mechanism for action, right? So you want to measure glycosylation of more proteins, and there are some successes in this way. I hope to talk about that next year, right? You want to measure things from more tissue samples using more technologies, better precision, put it to multi-layered omics, etc., start with functional screens. That's our way to, do it wide, right? But I think what I would really love to see is, so many of you, right, and I will not tell names because I couldn't stop, right? Many of you are doing different kind of, biology, right? You guys dig very deep, and the things you are seeing, you often achieve much better degree of consistency, right? And it wouldn't go over the whole genome or about, 500 diseases at once, right? But the degree of consistency and understanding is much deeper, right? I think the problem is not even that when I do these wide things, I see these inconsistencies. Inconsistencies is okay. What I really would like to see in the future is that this deep way and the wide way come together, because they almost seem to act often orthogonal, right? So the things I'm doing and talking about almost doesn't ring any bell with, with people who are doing very deep biology, right? And then we try to put it together. You really like, okay, how does this together? It doesn't really work, right? So I want to see more consistency between these two directions of exploring human biology. Okay? Right, thanks to the left people who did the work. So Bo is coming later today. He will be talking tomorrow about one of our works. Anna was supposed to come, but she couldn't. She was supposed to talk after me, so I'm going to spend 10 minutes presenting some of the major discoveries here. This also means that although I just went over time, I will not keep you from lunch. Okay, so thank you.